contribute to the success of this event. And as you told, actually, my focus will be mainly exactly on up and down slow oscillations, and how this up and down slow oscillation actually can display some metastability, contributing to the richness of the cortical dynamics when you move from the unconscious state to the conscious one. So when you move from anesthesia or sleep and wake up from this state being once again conscious. And as we are speaking about metastability, I think that a good starting point might be this canonical circuit introduced in spiking neural networks by Daniel Amit and Nicolas Brunel, and has been used to model working memory, so short-term memory. And the idea in this case that under Millfield approximation, a network of spiking neurons composed of excitatory and inhibitory neurons, like in this case, with strong enough self-excitation, synaptic excitation in a small subset of neurons, the foreground, can display nonlinear dynamics, and particularly attractor dynamics. And this is an example of what is happening whenever you change the strength of the excitation, the synaptic excitation, which brings to have a symmetric breaking, in which the network, when the coupling is weak, you have only one attractive state at the low firing rate. But if you increase this self-excitation, you will end up to have two stable attractors, one at the low firing rate and the other at high firing rates, which can be depicted by this Lyapunov function. This effective energy landscape has two valleys. And depending on the simulation, you will be driven to go in one of these two states, and you can escape from this metastable states. This is a canonical circuit because actually does not only describe the dynamics of a cognitive function like the working memory or short-term memory, but actually if you put together many of these canonical circuits, you can build a network displaying global transition that are has been successfully used to describe what is going on in cortical areas when other cognitive functions can be performed. And an example is shown here with this. This is a very classic paper by Shinoy and collaborator, where they look at the latent space of the activity in motor cortices when a monkey is performing a reaching task. And these are the trajectories, the gray curves. The trajectory follows by these neurons in an effective latent subspace when the animal moves from one behavioral state to another. For instance, when the go to appears on the screen and then the animal moves, and all the trajectories represent moving in a different state. And this can be described as a coordinated activity of many of the cortical circuits I showed before. And we did a contribution to this description, in which actually these trajectories are the result of a kind of chain reaction, in which these bistable networks are eliciting some switches in other, and so on and so forth. And so this is metastability in this sense, and there are plenty of other examples related to this kind of dynamical phenomenon, which in the end underlies many cognitive functions. But what I will focus on in this talk is a particular state, the one we experience every day when we go to sleep. And in this case, if you pick an electrode, you stick an electrode inside the brain. This is the classical picture coming from Steria, the work. They recorded intracortically from a cat, and simultaneously they also recorded the EEG recordings, which are the top traces. In the end, this is instead the intercellular recordings from a neuron in the visual cortex, if I remember, from a cat. And you see that this animal is falling asleep exactly at this point. And before this, you see that the spiking activity of these single neurons are rather irregular. the EEG signal is has small fluctuations and it is rather asynchronous. But when the animal falls asleep, you start to see very large fluctuations in the EEG signal in both hemispheres. So this couple of signals are coming from one hemisphere and this from the other hemisphere. So it's a global phenomenon involving the whole brain. And the whole brain at different scales. Indeed, if you have a look now at what is happening at the level of the single cell, you start to see a period of hyperpolarized state, which are named down state, in which the neurons are almost silent. And then they are followed by an activated state, the up state, or on state, as if you prefer, in which the membrane potential is depolarized and the neuron starts to fire again. These are the up state. And you see that this oscillation moving from down to up, are very much synchronized across the whole brain. And you can describe this kind of phenomenon having a look to the detailed time course of the firing rate from intracortical recordings. This is an example coming from the Mami Sanchez-Vives lab in Barcelona. This is a work we've done together. And you can see that whenever you stick an electrode with several contacts inside the cortex, you see that this up and down oscillation, this is a single cycle, both from the perspective of the local field potential and the multi-unit activity, which is a lamp signal telling you how many spikes are produced by a cell assembly nearby the electrode per unit time. You start to see that in particular in layer 5, it is rather apparent that there is a big increase of firing rate when this column is in the upstate. And if you zoom in and you average across several upstate, you see that this is the average shape of the cycle down, up, down, at the level of the synchronous state induced by anesthesia in this case. If I remember, this is ketamine anesthesia. And what is nice here is that if you have a look to the distribution of the multi-unity, so the firing rate of the cell assembly, you start to see that distribution is bimodal. And the residence time in the down and in the up, at least under this condition, anesthetic condition, are relatively regular. This is the distribution of the duration of the up, red, and the down states. So this is a very reminiscent of an example of a metastable state. And having a look to how to model this alternation between down and up, you can recover what I discussed at the beginning, because the canonical cortical circuit actually, thanks to the fact that you have a strong synaptic excitation, allows to describe very well the transition from down to up, because the transition down to up is not a kind of chain reaction induced by the fact that the coupling is strong. And this is what is happening in this small time window. Now, if you can imagine that these are the single neurons of a cell assembly we are sampling, and whenever one of them by chance start to fire, this red, it will deliver this activity through synaptic transmission to others, and then a chain reaction starts. This is the reason why you have this very stereotyped and sudden transition from down to up, which under mean field description, as discussed before, can be represented as a downhill pathway from the down state towards the up in this bistable dynamics. But if you look now at the other phase of this cycle, the one where you have up state moving towards the down, you see that actually an important player is missing. You need some inhibition in order to kill the stability of the up state in order to have such regular duration of the up and down states. Of course, this is not always the case. For instance, in other sleep, as I guess we will see later, the variability of these up and down states are rather large. But it is important in order to model this activity to introduce another ingredient. And the ingredient is a kind of self-inhibition, which represent the fatigue of this network. In this cycle, up-down cycle, where the blue means the firing rate of the pool and the red represent the level of fatigue, you see that you have a maximum fatigue at the end of the up state, and then there is a kind of recovery period during the down state. This red state variable is associated to the phenomenon of spike frequency adaptation. And this level of fatigue in the end under mean field approximation can be very well described as an inhibitory feedback. Let's say that this is the very rough representation of the dynamics of the frame rate of this pool of neurons I'm investigating. This is the input output gain function, let's say the Sigurd-Ricciardi gain function, which depends on the input currents. Input currents which are synaptic currents, mainly due by the activity of the network and due to the level of this fatigue. Stronger is this parameter G, which tells you how strong is the feedback, and easier will be to kill the upstate. This is an example, for instance, you have it down to a transition, and if the G, the strength of fatigue, is not strong enough, there is an adaptation in the frame rate, but this adaptation doesn't destabilize the upstate. This is not the case when you move from a condition in which you have a stronger adaptation. Indeed, stronger is the adaptation and the shorter is the upstate duration. This additional ingredients actually leads to have a different shaping of the Lyapunov function in such a way the upstates start to be unstable, and then you have a downhill descent from this valley to the other state. So this is a classical way to describe this phenomenon, that once you assume that the typical timescales of the frame rate dynamics is much slower than the adaptation timescales, as in this case, this is very well represented by a relaxation oscillator, where you have now 2 degrees of freedoms, which is adaptation on the x-axis and frame rate on the y-axis, and the adaptation slowly change the NLG landscape, the Lyapunov function of the system, in such a way you will destabilize and restabilize the up and down valleys, corresponding valleys. So this is the very simple way to describe this slow oscillation at the level of single unit. Okay. Once you have this 2 variable, actually, you see that this 2 degree of freedom are capable to describe also the different regimes this network is capable to express. And for instance, you can describe simply changing the strength of adaptation in this bifurcation diagram, the fact that you move from a burst suppression-like regime in which the activity is almost silent, and then you reduce adaptation and you start to see these sparse in time upstates, regular slow oscillation when the adaptation is weaker again, until you come up, when you cross the border, this transition and this state, which is reminiscent of the synchronized state of awake-like desynchronized state. So this is a very simple picture that captures all the richness offered by this state. But you have to consider that slow oscillations are a global phenomenon that involves all the brain. This is an example of recordings coming again from the Mavis lab, in which each row is a channel on top of the cortex. And this is the way in which the upstate occur. Upstate actually occur as waves. You can see here, these waves are how they occur. And depending on the level of anesthesia, actually these waves are shaped in a different way. In particular, when the anesthesia starts to be lighter, you see that the frequency of this slow oscillation is higher, and not only, also the pattern of this wave start to be more rich. This means that actually when you move towards the border that separates slow oscillations, low wave activity, and the awake-like state, actually start to have spontaneous activity which is richer. And this richness is described again, resorting to mean field theories, as a synchronized phase transition, in which again, the critical parameter is the adaptation. And in such a way you can move when you have this field of neurons connected together, showing silent state, bar suppression, regular, almost regular up and down slow oscillation in this case. And when you approach the critical point, the border between the yellow and the white areas, you start to see that there appears some long upstates, and the interval between upstates are more irregular. Of course, when you cross this border, you start to see the asynchronous irregular state, which is the one associated to the synchronized state leading to consciousness. Okay, and if you come back to this macroscopic level, at the level of the cell assembly, actually you see that this richness is characterized by something that is once again metastable dynamics. And this is an experiment in which the rats are anesthetized with ketamine. Again, we have a silicon probe in the visual cortex. We focus on the activity of layer 5 pyramidal neurons. And what you see on the right is the local field potential at different level of anesthesia. Here, anesthesia is ketamine, methamphetamine, and the fading out of this anesthesia leads the spontaneous activity you record from this cortical column, let's say, from a deep stage 1, deep stage 2, and then a light anesthesia level, which show a peculiar footprint. So when you are very deep, the oscillations are irregular, as in this case. Then, as the anesthesia lightens, you see that this oscillation starts to be regular. But at some point, at this point, when light 1 label can be observed, you start to see also another rhythm. This is the spectrogram of the local field potential. And you see that the regularity of the delta rhythm is associated to the width of this resonant peak. So shorter is, more regular is the oscillation. But the appearance of this second peak is telling you that there is another rhythm. A rhythm which is at the infraslow oscillation, as we say, and this infraslow oscillation are ranging at 0.5 or even less. And under this condition, you start to see this pattern. You see that there are slow oscillation and then irregular activity, slow oscillation, irregular activity. And the pace of the jumping from the synchronized and synchronized state is at this infraslow frequency. Okay, so this is the evidence. These are known as gamma bursts, are also observed in humans, and we will see that actually this is tightly related to what has been observed also under natural sleep. But to make the story short, this alternation between slow oscillation and the synchronized state is not only observed at the level of local field potential, but also at the level of multi-unit activity. So it's a local firing rate that displays this asynchronous activity, which is named microarousal, because this is very asynchronous, desynchronized as in the wake state, and the slow oscillation. So there is this alternation, rather regular. And we have been able to model and to predict some footprints. I will not tell you this because of the lack of time. Again, resorting to the bifurcation diagram I showed before. And in this case, what we changed was both the level of excitation on the x-axis and the level of adaptation, because these are a very good way to represent how the fading out of this kind of anesthesia is occurring. Okay, once you approach this critical point, the one separating the slow oscillation phase and the asynchronous wake-like state, you start to see that the slow oscillation are sparse, irregular, then start to be regular, and you start to see this alternation between the microarousal and slow oscillation. But why this? Well, this is because actually we are inducing the model an external perturbation, which is sinusoidal at the frequency of infraslow oscillation. And this is a very tiny perturbation, a tiny input we should provide to the network. Why? Because we are close to a critical point. And the critical point has the advantage to be maximally sensitive, having long-range correlation, and everything is in contact with each other. And so a very tiny perturbation can elicit a transition from two available states: a limit cycle, the slow oscillation, the relaxation oscillator, and the microarousal, which is an equilibrium point, a stable focus. Indeed, we confirmed this under near-field approximation. We found that this region where we have L2 is this case, is where you have, so we are moving in this direction. You have on the left only slow oscillation. So this is the tip at the bottom of the slow oscillation in the fine rate. And on the right, you have only one fixed point. Right, I'm almost finished. And you see that here you have a limit cycle coexisting with an equilibrium point. And this is the place in the bifurcation diagram where you can have this alternation between microarousals and slow oscillation. An autonomous system can be designed in order to produce this nested oscillation. And this is simply obtained, again, having the canonical circuit with adaptation I showed before, coupled with another element, which is likely modeling the brainstem, where is implemented all the machinery related to circadian rhythms. So the machinery that is governing the transition from sleep to wake and coming back to it, okay? So if you have a look to the activity and then to the current provided by the brainstem in this model, you see that whenever you have slow oscillation, there is an increase of this current. So there is a kind of accumulation of activity. And then you have a destabilization of the slow oscillation state, and such that only the microarousal is there. Under this condition, you have a relaxation of this current, and then you have once again the next cycle. So you have a bouncing, a nested cycle. So a slow cycle like this one, showing this long cycle, together with the very fast slow oscillation on this phase and the asynchronous state on this other phase. The properties of this cycle can be simply governed by changing the level of excitability, the external current you can provide to the cortex. Indeed, when this input, excitable input, is strong, you are making more stable the microarousal. And this is the case when you are approaching wakefulness and the microarousal are longer. On the other hand, when this level of excitability, the degree of wakefulness is weak, you start to have the opposite, a very long period of slow oscillation, a very short period of microarousal. This is just to tell you that, again, this is a very nice example. As I told you, from the experience, we also tested this hypothesis of the existence of these two attractors, finding many proof of the reliability of this theoretical description, that is telling you that there is another kind of metastability. So there are several metastability that are differently expressed depending on the global states of the brain. And in this case, we found that this alternation between slow oscillation, here we have the EEG patterns coming from a human brain, are interspersed by microarousal naturally. And by Parino Tersano here in Italy, they introduced the concept of cyclic alternative pattern. The cyclic alternative pattern is the proof that the sleep is unstable. And this instability is due to the fact of this competition between a limit cycle, as I described to you, competing with a stable focus in this simple matter. And just to wrap up and to tell you which are the key messages, the take-home message is, okay, the metastability of cortical networks resorting to a generalization and enrichment of the canonical circuits which we used to model perceptual decision or short-term memory actually has to be enriched with an additional current, which is the potassium current associated to spike frequency adaptation or short-term depression, so short-term synaptic depression, okay? And thanks to near field approaches, we can describe a very simple bifurcation diagram that allows us to describe this transition from sleep to wake or from deep anesthesia to wake. This transition is a specific phase transition, which is the synchronized phase transition. And at that point, you have a subcritical of bifurcation where coexist these two local state, the slow oscillation and the microarousal. And I conclude, of course, thanking all the collaborators that contributed to the work I showed and the funding coming from the Human Brain Project, and of course you for your attention and Tilo for the organization of this nice workshop. Thank you.
Thank you, Maurizio, for this very nice talk. There's time for only one very short question. You can just unmute yourself if you have a question and directly speak into. Yeah. Otherwise, I would like to know about a little bit more about the model. So this is a rate model, or is it a network model of integrated fire neurons, or is how do you add noise? Because sometimes I see some stochasticity in the traces. And I wonder whether this potential that you show is more like for illustration, or whether you can indeed have a potential system.
But thanks for
was
the
already
question.
a long question, but
yes, it's a long question, and actually
it's
a very intriguing question, because as you can understand, that as we are speaking about metastability, the role of noise is important. And actually, the way in which, so these are mean field rate models. We introduce the finite size fluctuations in a very simplistic way. So these fluctuations are related to the finite size of the network. Let me tell you that the statistics of the residence time in the slow oscillation and microarousal state should be strongly related to the size of this fluctuation, so to the size of the network underlying. So let me say that also we are expecting to see phenomena like stochastic resonance, and in particular, this might be also a way to estimate the size of the local circuits in some way.
Okay, so maybe because of time, we need to move on. But thank you again. So now, Bastian Pietras is the next speaker. Can you please share your screen?
Sure. You can see everything.
Yeah, perfect. Yeah. So thank you, Bastian, for being here. So it's my great pleasure to hear your talk. Yeah, so Bastian currently works in Barcelona with a Marie Curie fellowship together with Ernest Montbriot. Before that, he worked as a postdoc in with myself and with me, and before he worked in Amsterdam together with Dafatshofer on Kuramoto oscillators. Okay, so Maurizio, we hear you. Okay. So Bastian will talk about a mesoscopic description of hippocampal replay dynamics and metastability in spiking neural networks with short-term plasticity. So please.
Thanks very much, Thilo, for the introduction. Thank you to all the organizers for giving me this opportunity to talk about this joint work together with Thilo and also with Valentin, who's working at the EPFL in Switzerland. And I'm talking mainly about a mesoscopic mean field model for spiking neural networks and how metastability arises in these kind of networks. As we've heard before by Maurizio, metastability, or one of these important or key factors for metastability, is some activity-dependent fatigue mechanism. This can be either like via spike frequency adaptation, mediated by calcium-activated potassium currents, and that's basically the focus of our work, of my talk, via short-term synaptic plasticity. So it's sort of like indicating how many neurotransmitters can be released after an action potential. And the other key factor are basically fluctuations, and here I will focus on finite size fluctuations. Okay, so let's start with short-term synaptic plasticity. Like one of the seminal models by Zodix Markram was, if we have like a neuron, like here, that elicits spikes, then usually this presynaptic spike train is modulated by facilitation and depression variables that amount to like the number or the amount of neurotransmitters that can be released after that action potential. So whenever a neuron elicits like a spike train, what actually comes out or comes as the input to a postsynaptic cell is pictured down here. So you see if like neurons are firing, or like if the spikes come rapidly one after the other, then the effective spike or the effective spike train decreases, and then after a time of no spiking, then the spike strength, the input can be increased again. So this is one neuron. Let's have a look at what happens if we like simulate a full network of those neurons. So here we have like 100 neurons. There's no fluctuation. They are all coupled. They're only like, the only thing that's in there is they have like this short-term synaptic plasticity. And we see that there are like different states visible in this neuron. Like for instance here, like all of these neurons kind of spike together. So this is like a population spike. Then there's a period of silence in between, and again some other neural activity. So every dot in here means this is one spike of that neuron. And yeah, we have like exactly these population spikes, and sometimes, not after every population spike, but sometimes after such a population spike, we can enter in an up state. Like here we have like the population activity or the mean firing rate of our population, where we see like we have an up state, and between population spikes, then we have down states. Another important factor here is like if we increase the network size from like 100 to 500 neurons, like this is this orange line, then basically the firing rate, the mean firing rate, the mean population activity remains silent, and there are no transitions between these metastable states. So the goal of this talk will be to derive a mesoscopic mean field model in order to better understand metastability that arises from the interplay of a slow fatigue mechanism, here short-term synaptic plasticity, and the finite size fluctuations inherent to our network. For simplicity, I only like look at short-term depression. So we only look at like the depression variable and forget about the facilitation variable. So here, H, just to guide you through the equations, H is, or HI is the input potential that every neuron I receives. There's a common input mu. Then we have like this leak term, tau is like the membrane time constant. And then we have here like an interaction, like an all-to-all interaction term, all-to-all coupling term, where we have xj's, which are basically the available synaptic resources at a presynaptic neuron. And sj is the spike train. It's like a delta spike train that you see here. And in general, we have like u naught, like a mean facilitation variable times xj times sj, which is the modulated spike train that we saw before. So this is basically this u naught si, what you see here, this modulated spike train, what comes into that. Okay, I also have to tell you, okay, this si is like the delta spike train, which occurs, or where every spike is modeled as like as a time inhomogeneous Poisson spike with intensity f of h. So this f of h is like a smooth threshold linear transfer function, and you can think about that as like some expected mean activity. All right. So how can we go from here, from this microscopic network model, where we consider like n neurons, to a mesoscopic network model describing like a mean field model? Importantly, in this mean field model, we have like to consider two sources of stochasticity that shape the fluctuations of our finite size network. So one is really the spiking output, like this shot noise stemming from the Poisson, like from this Poisson spike train. And the other one is that we have like heterogeneous depression due to the individual spiking. So it's like this xi variable, like the synaptic resources that will like differ over our whole network. So in order to describe our mesoscopic mean field model, we have to take into account both of these kind of sources of stochasticity, and they, as I will show, they will shape the fluctuations of our network. Okay, so like a general idea is we introduce mesoscopic variables, h here and x, which basically are the means over the hi's and the xi's. So we can write that down. We end up with these sort of mean field dynamics, but the problem is that we have these sums in here that still depend on the individual neurons. And the basic idea to overcome these sums, to introduce them or to replace them by mesoscopic variables, is to make use of a chemical Langevin equation. So we have a look at this integral down here, and the time or like the increment during a macroscopic infinitesimal time step in the limit when we have like a lot of neurons. So this term here can be described by a Gaussian with mean and variance. And while usually heuristic macroscopic models forget about this this variance, we here make, well, explicitly account for the variance as well. We see the variance here scales with 1 over n. So we really have like these finite size fluctuations. And on top of that, we have like this factor Q in here. Q is basically the second moment of all the xi's. And yeah, in order, like our mesoscopic mean field model will also describe the second moment of the xi's of the resources. So to spare you like a lot of the details of the derivation, in the end, we end up with this mesoscopic dynamics for our mean field H, mean field X, and for the second moment Q. And as you see, like the only term where we have like the noise term is that this noise is basically shaped by Q and also by f of H. So we have like some kind of multiplicative noise that shapes the dynamics. So interestingly, in this diffusion model, like a first step is we can go to the limit n to infinity. And if we take n to infinity, the noise vanishes and we end up with I call it with the macroscopic dynamics, or I call it this deterministic backbone. And usually this deterministic backbone can give us like hints at like all the dynamics that are relevant for the actual network behavior. So for instance, just looking at the deterministic backbone, like at this two-dimensional system, we can apply dynamical systems theory, draw the null clients in this HX plane. We can determine the stability of the fixed point. So in general, we have like here a stable fixed point. The input potential is quite low. The depression level is quite high. So this is a stable fixed point. Then we have like an unstable fixed point here. And in between this diamond, this orange diamond is a saddle point. So we can also have a look at the saddle manifolds of our saddle point. The stable saddle manifold is the separator X. And if we have a look at the unstable manifold, then it gives us the orange dotted curve. And this is interesting because if we really like simulate our photo mesoscopic model, then the stochastic neural trajectory will follow this unstable manifold. Once we are like on top, like above the red separatrix, we follow the unstable manifold and make this excursion back to our unique saddle point. But because of these finite size fluctuations, at some point we will be kicked beyond or kicked across the separatrix and then run along the orange dotted curve along this unstable manifold back towards the fixed point, to this low activity fixed point. So if we look at like a trajectory over time, then we see in this H dynamics of our input potential really these spikes, these population spikes, and the X variable goes like that. Okay, so here we have like a nice population spike diffusion model. How does this compare to our microscopic network dynamics? So in order to compare the dynamics, we can run simulations for as few as like 30 neurons in our network, have a look at like a qualitative look. This looks pretty nicely. And also if we look at the frequency of our dynamics, then we see a very good match. Also the interspike intervals, like if we only look at the differences between like these population spikes and like compute the probability density function, then we see like a very good fit between the microscopic and the mesoscopic model. There's like a little bit of divergence here in the peak, but as soon as we increase the number of neurons, they go to 200, then we have like an almost perfect fit. So the larger the network size, the better our diffusion approximation, the better our mesoscopic mean field model. Okay, now let's change slightly the parameters. So changing the parameters makes this upper fixed point to become stable. Before it was unstable. Now it's stable and it lies inside an unstable limit cycle. So as before, whenever we have like a trajectory starting close to this low activity state, then finite size fluctuations will kick us across the red separatrix. We make an excursion, like running along the unstable orange dotted manifold, subtle manifold, and on the way back to our down state, we come very close to the unstable limit cycle, which is the boson of attraction of our stable fixed point here. And finite size fluctuations can kick us inside it. We try to run into the upper stable fixed point until other finite size fluctuations will kick us out of the limit cycle, also out of the boson of attraction, so that we run back to our stable down state. So this is exactly this transition that we have here. From the down state, we go to the up state. We have like this kind of ringing behavior, which, yeah, well, the frequency that's in here corresponds to the stable focus of our stable up point, and then we can switch back to the down state. So that is basically the dynamical insights, the dynamical mechanisms that is hidden behind our mesoscopic mean field model. And again, we can compare this dynamics, what we've seen in the mean field model, with the microscopic networks here for like 100 neurons. Again, it looks like qualitatively looks very good. Also quantitatively looks very nice. But yeah, there's slight deviations here, and especially if you look at the histogram of our potential, then we see that there are some stronger deviations that we had before. And but the nice thing here is that we can make up for these deviations because the discrepancy here that we see between our mesoscopic diffusion model and the microscopic network is that this discrepancy is due to the spiking output, this Poisson shot noise. And by going a step further and introducing like a hybrid model where we explicitly model spiking output as Poisson shot noise, we can get like a perfect fit. The dynamics will look like that. So here this dz dt is then the Poisson noise, but I don't want to bore you again with the details. Yeah, the result is that this hybrid model really captures network dynamics for arbitrarily small network sizes. So even here for n equals 30, we have like a perfect fit, where before the diffusion model wasn't as strong as it was. So as an intermediate summary, yes, we can derive a mesoscopic mean field model for like spiking neurons with short-term synaptic plasticity. It's important that this is a bottom-up approach. And the accuracy of our diffusion model depends both on the network size, but also on the dynamical regime. So when we have like this transition between up and down dynamics, there it was important that we really concentrate or focus on the particular realization of our Poisson output. And as an add-on, this mesoscopic mean field model, this new mesoscopic mean field model, allows for new insights into hippocampal replay dynamics, and that's the part that I want to focus on the last couple of minutes.
You have 7 minutes.
Right. Okay. So for the replay dynamics of place cells and hippocampus, usually if we have like a rat running around a corridor, then it's known that we have like kind of neurons here in different colors that code for particular locations of our rat. Interestingly, once the rat kind of stopped running through this corridor and it starts dreaming about that, then we have like this replay. So without being in this exact corridor, the mouse or the rat like rethinks of this trajectory, and this is called replay. So we have like this kind of sequence of active patterns now going in reverse. Sometimes this can also happen in the reverse direction. This is called pre-play. So this is a nice cartoon, but it also exists in real data. Usually people measure data from CA1 because it's easier. To model that, it's better to really look at hippocampus area TA3, because there we know that we have some sort of recurrent connectivity, so that we can kind of store we can store an environment in the coupling of different neurons or different populations within the circuit. So one of the important models was by Romani and Sodix 7 years ago, where they considered a circular environment, sketched down on the left. We have they took 100 heuristic mean field models, coupled them via this Mexican head coupling functions. And what they found was, with the short-term plasticity, that you see kind of metastable activity here on the average activity, averaged over all 100 populations. And from time to time, you see these bursts. And even better, if you zoom into one of those bursts, then you see that actually there's trajectories running across the place position, the place field position. So you run into either forward or kind of backward direction. Then they did a lot of statistics, but again, for the sake of time, I try to skip that. So we try to model exactly the same kind of hippocampal dynamics with our model. So instead of using Romani and Sodix's deterministic fine rate model, we used our mesoscopic description. So in the middle, we have our macroscopic simulation, the left, the mesoscopic simulation. We also see this kind of bursts. So there's a good fit between mesoscopic and microscopic simulations. It also fits to Romani and Sodix results. An important difference, however, is that in our case, it's a pure finite size effect. So as soon as we increase the number of neurons per population, going n to 5,000, then we see the zero line, no activity, no replay dynamics at all. From the statistics here, there's also a good fit. It looks qualitatively the same. So what are actually the differences? And the differences, just to tell you a little bit more, so if we look at the sequence of these kind of replay dynamics, so if we look at the sequences of replay dynamics, sort of the intervals between two bursts, then in our model, in our mesoscopic description, here we have a kind of a unimodal Poisson-like interburst interval distribution, and there's hardly any serial correlation, so of between consecutive forward or backward replays. Whereas in Romani and Sodix's deterministic model, you have a bimodal distribution of these interburst intervals and also strong serial correlations. Comparing that to experimental data, here are some examples, it looks like that experimental data really shows a trend that we have a unimodal distribution, which kind of makes, supports our idea that it's kind of a finite size effect. But it's also possible that a combination of fatigue or fatigue mechanisms and finite size effects together form this metastable hippocampal dynamics. And as a last point to end is we also tested one of the other hallmarks of hippocampal replay dynamics, namely storing different environments in the recurrent connectivity matrix. And then you see that you have replay of just one environment. So here's one environment, environment 1 is active, then environment 3 is active, whereas the other two are different. And the results basically are that in our finite size model, or in this finite sized induced regime, the replay dynamics are way more random than in deterministic models. And more information about that is in our archive manuscript So yeah, thank you very much. And if you're interested, have a look at the archives.
Thank you, Bastian. So yeah, we have time for 1-2 questions. Please just either unmute yourself and ask a question directly, or write something in the chat. At the moment, there's nothing in the chat.
I had a question. So, can you hear me? Yes. Okay.
Who's speaking? Sorry.
It's
okay.
me.
Now
Yeah, it's me.
yeah.
I didn't turn my video on. Sorry.
Oh.
Thank you for the great talk. I was wondering, in the simulations that you had for the Matsucado model, for the place cells, where you see this nice sort of spatial pattern. So do these come about then spontaneously from what kind of stochastic input is put into the model so that these come about?
There's no stochastic input at all. So it's like really, well, the only stochasticity in there is that we have like stochastic firing, but there's like the external inputs are deterministic. It's all receive the same input. It's kind of like stationary. So there's nothing that triggers single events. Like all these bursts that you see here are kind of like really metastable states where where kind of like the finite size fluctuations of the network. Well, it's not really finite size. It's kind of like high dimensional, right? I mean, like finite size fluctuations are like this very high dimensional dynamics. Well, leads to these kind of high activity states.
All right, that's very nice. So what was there's the same input to all the
same input to all neurons. No external noise. Exactly. It's all inherently generated.
And are those periodic inputs or?
No, it's like stationary.
Oh, okay. All right. That's pretty cool. Okay, thank you.
Okay, is there another question? Otherwise,
there are a few, I think. I think
sorry, maybe I.
And one other person had their hands raised on the Zoom before.
Okay. No, sorry, I didn't see. So I see there's several. Excuse me. So Gabe, you have a question. So maybe a short one, because we have also to move then, or further.
Sure. Yeah, I had I guess one really quick question and one maybe slightly longer one. So the really quick question is that like 30 is remarkably small. So does the hybrid model, does it fail for 5 neurons or for 1?
Oh, good question. I didn't test 5. I didn't test 1. It's like it worked for 10 and 20, or for 20 it worked remarkably well. Like, and then the question is, why do you need it for 5 neurons? Then you can also.
Yeah. My second question was, so here you suggested that kind of the hippocampal replay was due to these kind of finite size induced network reactivations. And my understanding, at least, is that you have fluctuations in these networks through finite size because of this 1/n coupling, right? Like without the 1/n coupling, if you had stronger coupling, right, then you might not lose fluctuations in the large n limit. Or you could also think about other kind of intrinsic sources of noise in these networks or variability, like stochastic vesicle release, right? Like the microscopic model that underlies this short-term depression. So do you have any thoughts on comparing those like different potential sources of fluctuations on the statistics of these reactivations?
Well, honestly, I have no idea. I haven't thought about that, but it's a really great question. It's like really, really interesting to see, okay, if you make these XIs and like here in those dynamics, if you make like these XIs like stochastic on top, whether that gives you like more dynamics. I mean, if you like just going
maybe
from the microscope,
maybe you can discuss. Sorry if I break you. Maybe you can just discuss
Sure.
later together, because we are a little bit late in time. So I would like to stop here and move to the next speaker. Sorry. So yeah, thanks again, Bastian. And the next speaker is Daniel Liebenstein from McGill. So yeah, Dan did a bachelor in biochemistry with a minor in physics, and then did a master in biophysics. And then he started to study the neural dynamics during sleep at NYU, together with John Rinsel and Georgi Buzaki, where he did very interesting work on up and down states and the different types of dynamics, and how to infer the different types of dynamics from data, from population data. And today, Dan will talk about excitable dynamics of the sleeping brain.
Cool.
So please,
thanks, Tilo.
I'm happy that you're here.
All right, it's cool. So I'm going to talk about some of the work that I did with John Rinzel and Yuri Buzaki at NYU, and then a little bit of follow-up work that I've been doing with Adrian Pietras at McGill. Let's see. Okay, yeah. So basically, one of the earliest observations in neuroscience is that the brain does the most interesting stuff while you're asleep, right? So this goes back to the 1930s, when Alfred Loomis would have his friends wear his new fancy EEG set when they took naps at his house. So he would have his, New York City friends come to his house and just take these naps and record whatever happened. And so what he noticed is that when they fell asleep, you start seeing these large fluctuations in the EEG signal, right? So these are larger than basically anything you see during wakefulness. And importantly, as they fell into what he called deeper and deeper sleep, the fluctuations got bigger and bigger. And so let's fast forward now 80 years. And so now we know that these large deflections from the EEG correspond to synchronized alternations in neural populations in the cortex between up and down states, right? So I'm showing here about 6 seconds of data recorded by Brendan Watson from the frontal cortex of naturally sleeping rats. And on top, you can see the LFP, or the intracranial equivalent of the EEG. And below, you can see the spiking activity of 62 simultaneously recorded neurons in the cortex. And notice that every time you have one of these large positive deflections of the LFP, you have silence in all of the neurons that we've recorded nearby. And we see this on the population level. Now, when we look at all of the neurons in the data set, when you align their spiking to the positive peak in the LFP, you see basically that all the neurons stop spiking. So these are our up and down states, which correspond to the slow waves in the LFP. But there's this big open question, which is, what are the underlying mechanisms of these slow oscillations? And so this is kind of what we wanted to tackle during my PhD. And so fortunately, we didn't really have to start anywhere near from scratch. And that's because these synchronized up-down alternations are really the default mode of cortical tissue when it's offline. So they're not only seen during non-REM sleep, but you also see alternations between up and down states during quiet wakefulness and sensory cortex, as well as under anesthesia, and even in slice and cortical culture preparations. And so because of all this work, especially in the more in vitro preparations, there's been a lot of good modeling work, and it's all kind of come down to this canonical mechanism, which is that these alternations come from the interplay between recurrence and adaptation, as Mauricio also talked about earlier. And so basically what ends up happening is during the upstate, recurrent excitation maintains activity in the network, but also you have the buildup of adaptation currents, which eventually are strong enough to bring the network into a down state. The network then is able to recover from adaptation because no one is spiking. And then eventually the population is able to bring itself back into the up state, either due to the loss of this adaptation current or the effect of noisy fluctuations, which set off this chain reaction again. And so what we wanted to do was to kind of build on this extensive modeling work, which has models a varying range of detail, and use it to model what to match to what we see in our data during natural sleep. And so what we decided to do was to kind of abstract this simple or this model into as simple a formulation as we could, which hopefully would allow us to study the behavior of the mechanism and how it relates to the effective parameters. And so we did this with an adapting Wilson-Cowan model. Basically, the model represents the activity of a neural population. It has input from an external source, as well as recurrent connections in the network, and then subtractive current due to the effects of adaptation, where adaptation is turned on by the rate of our population and acts to self-inhibit the population, and it has some slower time constant tau a. We have sigmoid steady state functions for both the adaptation as well as the population input output function. And so what we find when we do an analysis of this model is that basically we have 4 distinct regimes of alternation dynamics, which I'm going to show you 2 ways. Once on the left with the phase plane, right? So trajectories, the dynamics of the model are represented as trajectories in the phase plane. Fixed points will be these solid circles. And I'm also going to go show you the time course on the left, where the dynamics of the population and the adaptation are shown over time. And so the first regime is an oscillatory regime, in which we have a limit cycle around an unstable fixed point. And in the time course, basically the population alternates between active and inactive states. So regularly alternates between up and down states, during which the population goes back and forth between this right branch of the R-null-Klein and the left branch of the R-null-Klein. We also have a bistable regime in which we have 2 stable fixed points. But of course, these are metastable because we have noise in the system and we're close to these saddle points. So as long as the noise is big enough, we can be kicked back and forth between these 2 stable fixed points, and the trajectories or the time courses will show these alternations basically between an active and inactive state, during which adaptation is active and inactive correspondingly. And then finally, we have 2 regimes which are excitable. So in each of these regimes, we have a single stable fixed point, but again, you're close to the saddle point. So noise can kick you over into the right or left branch of the R-null-Klein, before which you kind of cycle around and come back to the stable fixed point. And so in the time courses, the way these look is we have most of the time here in the excitable down regime, most of the time is spent down. But every once in a while, you could see that you have these transient excursions to an upstate, which are rather stereotyped. And so this is the trajectory in the phase plane going around the right branch of the R-null-Klein before it comes back to the stable fixed point. And the excitable up regime is basically the same thing, just the mirror image, where now the up state is stable in the stable point. Adaptation is tonically active, but then every once in a while you can get kicked over into a down state. Adaptation inactivates and eventually brings you back to the up state. And the reason that we have these 4 different regimes is basically because the up and the down state corresponding to the left and right branch of the R-null-Klein can either be metastable, right? So they can have a stable fixed point, which can give you noise induced excursions to the other branch, or they can be transient, meaning that there's no stable fixed point and you only need the effect of adaptation either turning on or turning off before you transition back to the other state. Okay, so the question we first had was, how do the parameters of the model determine which of these regimes you're in? And so we answered this by looking at a number of different bifurcation diagrams. I'm going to show you the bifurcation diagram with respect to drive and recurrence. And these are going to tell us where the transitions between the regimes happen in parameter space. So this is the IW bifurcation diagram. It has this configuration with the crossing of two cusps and a hop bifurcation. And so the result is you have this bistable domain at high recurrence, an oscillatory domain at kind of intermediate recurrence, and that 3 fixed point configuration of the excitable regimes with 1 stable fixed point kind of have this limited area in parameter space. Oh, and this is just to remind myself that if you look kind of back in the old bifurcation literature, this configuration is called a butterfly catastrophe, which is kind of a fun fact from history. But what does this actually teach us about up and down states? Well, first, that up and down alternations really require some sufficient amount of recurrent excitation. And they appear in this codimension 2 hop bifurcation, which we can actually in the model solve analytically. And we've mapped out where it occurs. From where you can see here. But the crucial thing is that you need some sufficient level of recurrent excitation to maintain a self-maintained upstate in the population. And then the other thing is that if we look at the fixed points as a function of drive for different level of recurrence, basically this is the effective input-output curve of the population, right? So how does the steady state rate depend on the level of drive? And this can basically take 2 different configurations. It can either be oscillatory centered or bistable centered. And we have a transition here where the cusps cross, which again, we can solve analytically. And it comes at some intermediate level of recurrence where you transition between having oscillations to having bistability. And then so the final thing is that despite the fact that we had this, we only have the excitable regimes in this tiny area of parameter space, when we actually run the simulations, we find that the excitable regimes extend pretty far out into parameter space away from the butterfly. And so what I'm showing here is the results now of many simulations of the model and the mean durations of both the up and the down states, where red indicates longer ups, blue indicates longer downs. And so you can see is that as we increase the level of drive, the up states get longer and longer. And crucially, we're able to detect up and down states, so bimodality in the rate, in pretty wide range of parameter space. And so outside the butterfly, what you see is that we have long up states on the right, long down states on the left, and more importantly, that the coefficient of variation of the upstate durations is very long over on the right, while the coefficient of variation of the downstates is very short. And we have the flip side on the left side of the butterfly, where the down CVs are very high, so the downs are regular and long, and the ups are short and regular. And so this is basically happening because what you have next to our bistable and oscillatory domains in the input-output diagram is a metastable configuration where even though you only have a single stable fixed point, because you're close to the either this hop or saddle node bifurcation, you're able to get these single excursions out to the opposite state, which is a form of metastability due to being close to this bifurcation. And importantly, what this means is that we can actually use the up-down duration statistics as a signature for which dynamic regime we're in. So notice here that in the oscillatory regime, your up and down state durations are all short and regular, while in the bistable regime, they're both long and irregular. And in the excitable up and down regime, one of your states, so in the case of the excitable down regime, your down state is longer and very irregular, while your up states are very short and regular. And so that basically, if we're to look at the up and down state durations, we can map that back to the model and say, where in the model do we see up and down state durations that match what we see in the data? Okay, so what do we actually see when we look at the data from our naturally sleeping rats? So here's the 6 seconds of data that I showed you before, and the thing that should immediately jump out to you is that the down states are, as a rule, very short compared to the up states and all about the same duration. And this should remind you immediately of one of the excitable regimes that I showed you before. And when we quantify this over the data set, we see that that this basically holds over all the recordings, right? So the up states are long, they all have a CV of about 1, while the down states are all short and have a CV of around 0.3. And so when we now go back and directly match the duration distributions to the data, so this plot shows the similarity or the KS distance between the upstate and downstate durations in the model and the data, we see that we have this band of high similarity between the model and the data in this excitable up regime, close to the onset of bistability or oscillatory dynamics. Okay, so you might remember now that I mentioned something about deeper sleep in the human EEG with more and larger slow waves as sleep gets deeper. And so we actually have something similar in the rodent, which we can quantify by power in the delta band, where deeper sleep has higher delta power. So the question we had was, how does this depth of sleep match with the model? And so to connect with the model, we again looked at the up and down state durations now as a function of delta power. And so notice that so this shows the upstate duration distributions as a function of delta power. And notice that while the downstate durations always stay about the same, regardless of delta power, what ends up changing is the upstate durations get shorter and shorter and slightly more regular. And so when we play the same game that we played before, basically matching these distributions from the data to what we see in the model, we see that as sleep gets deeper and deeper in the rodent, the best fitting parameter domain in the model gets closer and closer to the Hopf and saddle node bifurcations. And in some cases, so this would be some recordings that we have, it actually enters the oscillatory regime. So in these cases, the deepest sleep is actually an oscillation. Okay, so what have we learned so far about these excitable dynamics? First, that up and down alternations come in a few different flavors or different regimes based on the stability or transience of our up and down states. Second, that the neocortical slow oscillation is actually not usually an oscillation, but it reflects an excitable regime with a metastable upstate and a transient downstate. And so this means that the sleeping brain uses kind of this cool combination of noise and metastable dynamics to produce slow waves. And what I didn't show you is that actually we can fit the hippocampal data with this model too. So the hippocampal sharp waves are actually also well fit, except in this complementary excitable regime where the downstate is stable, but noise can kick you into transient upstates or sharp waves. And so for more, you can see this paper in Nature Communications with Yuri Buzhaki and John Rinzel. But I want to step back a little bit and point out that if we actually want to understand the mechanisms of slow waves, there's kind of this glaring open question, which is what's adaptation, right? We have a good sense of what recurrence is. Neurons are connected to each other and that provides this positive feedback. But we've modeled this adaptation pretty abstractly, right? Basically, it can be anything that's slowly turned on by neural activity and acts to reduce neural activity. And this has been modeled and also shown experimentally. There are a number of different ways that neurons do this, right? There are things like voltage-dependent potassium currents, sodium-dependent potassium currents, short-term synaptic depression could even act like this, as well as GABA B. And our model can't really distinguish these in the sense that all of these different sources of slow negative feedback would have something like the excitable up regime that we see in the data. But I kind of thought at this point that this is where this project ends, right? We've done our job. We've laid out at least a general theory of the dynamics and its possibilities, and now we can kind of pass it off to experimentalists, and they can go figure out which of these sources is actually adaptation that's producing the slow waves. But then I started my postdoc with this guy, Bastian Pietras.
5 minutes.
And he's awesome, perfect, thanks. And he says something to me around my first week in lab, which is that he has a student with some cool results that they can't really explain, and he was hoping that my model might be able to help. And so what he was talking about is that his student Dhruv was looking at some data collected by Adrian, in which they inserted a linear probe into the head direction cortex and were able to record the whole length of this cortical region from the dorsal to the ventral area. And what they found is that when they look at the data during sleep, and they look at the slow wave, so this shows an example slow wave or down state, the up to down transitions were synchronous, but when they ordered the cells from dorsal to ventral, the dorsal cells basically always seem to spike early.
Иван Купала. Зона поздравляет.
And so basically what they have is this traveling wave, but it's a unique kind of traveling wave where it's not really the delta wave that's traveling, but the down to up transition. So the return to the up state travels. And so we're able to quantify this when we look at the up onset delay, so the delay of spiking as a function of depth, we see a nice correlation. We only see this when we look at the down to up transition and not, at least on average, in the significant correlations in the up to down transition. And we can reproduce this when we look at the gamma power, so a good proxy of spiking aligned to the slow wave. You see that basically all the channels from, or a subset of channels from dorsal to ventral tend to fall into the down state at around the same time, while the return to the up state starts with the dorsal channels and travels to the ventral channels. So how might our model be able to explain this? Basically, what we have in the model is a number of different parameters that might be able to shape the down to up transitions. And we can use that to ask, okay, do these dorsal channels behave as if they're more excitable? Do they behave as if they're more recurrent? What in our model might explain this traveling wave? And so what we're going to do is build a spatial gradient into the model with a variation in one of these parameters along the gradient and see which parameter can result in the observed dynamics. So we started with excitability based on some observations that cells with higher firing rate tend to fire earlier in the upstate, thinking that, okay, maybe these cells are more excitable. And so this shows a simulation of the model now with a gradient in excitability from a dorsal to a ventral population. And when we align the activity in the model to the up to down and the down to up transition, we actually find that this excitability gradient isn't able to reproduce the data, right? So the more excitable or dorsal population not only goes up sooner and travels, so that does match the data, but it doesn't go from up to down synchronously. So it also stays up longer at the end of the upstate. And so it actually turns out that none of the gradients in the model are able to produce the up-down asymmetry. In all cases, you get an up to down transition that isn't synchronous, which was kind of the really key feature that we saw in the data. And I've only showed this with a single parameter example, but when we do a sweep through parameter space, we see that this is the case. So at this point, we kind of took a step back and say, that's a little disheartening. What can we do to our model to capture this? And so I was, awesome, thanks. I'll go through this really quickly. Just to say that I was reminded of something I discussed with John during my PhD, which is that a hyperpolarization activated inward current might act as a functional adaptation. So this is a property by which when you hyperpolarize cells, they tend to depolarize. And so in the model, this is basically adding a double negative, right? So instead of having something that at high input inhibits the network, we have something that is inactive at high input, but at low low rate activates the network. So these can effectively act as the same thing, even though one is an excitable or an excitatory current, one's inhibitory. And we can, with this rectifying current, also produce excitable up dynamics, just like we see in the model. And when we build in a gradient now of high IH dorsally and low IH ventrally, we're now able to reproduce what we see in the data. So we have a synchronous up to down state, but the down to up transition travels from the dorsal population to the ventral population. And this is basically because now we have a configuration in which the dorsal and ventral populations are affected the same during high rate, so during the up state, but during the down state, during low rate, the dorsal populations activate this H current stronger, so they return to the up state earlier. Okay, so conclusions. The interplay between recurrence and adaptation can give 4 different regimes of alternation dynamics, 3 of which are metastable, so we have a bistable and 2 excitable regime. The slow oscillation during non-REM sleep best matches one of these excitable dynamic regimes. So it has a metastable up state with a transient down state. And what we find when we look at the head direction cortex and the fact that we see this traveling up state onset, it suggests that actually the H current might be the responsible player for downstate transients rather than traditional adaptation currents. And so I just have to thank my PhD advisors, Yuri and John, Brendan, who collected the data for the first half of the talk, my postdoc advisors, Adrian and Blake, Adrian, who collected the data that I talked about in the second half of the talk, and Drew, who I've been working with on the analysis of that data. And thanks, Thilo, for organizing this symposium, and everyone for your attention.
I'm muted. So yeah, thank you very much for this beautiful talk. And yeah, we are already a little bit late, but maybe a very brief question, or one very brief question, if there is. Otherwise,
I think Mauricio has one.
All right, so Mauricio.
Very fast. Beautiful talk. I like it very much. Can you exclude the possibility that this synchronization is coming from outside the cortex?
No, you can't really. And yeah, I mean, I guess if you have your upstate is stable, and here, let's go back.
If it's a long answer, please, maybe you can also discuss after Luca's talk. So
I don't think we can exclude that, but let's talk about what that would mean, because once you start thinking about the input, you know, things get complicated, as you know. Yeah.
Okay.
Thank you.
Yeah. So let's move further to the last speaker of this mini symposium, given by Luca Matsucado. Luca, you can share your screen.
Hello. Yeah.
Hi.
Hi.
Hi. So Luca, also a physicist by training, actually a particle physicist who switched at some point to neuroscience, and he did a lot of work on metastability in the cortex, both with mean field theories and data. And yeah, so I'm happy to see you and hear your talk. Where are you? Are you in Oregon?
Hi. Yeah, thank you. So I'm actually in Aspen, Colorado, at a workshop on learning complex systems of behavior and metastable dynamics.
Anyway, it's very early in the morning for you now.
Yeah, I wake up at 5 a.m., but it's okay. So I have a problem with Zoom that I have to re-log in to share my screen. Sorry about that, because I just got a new laptop and I'm still troubleshooting. Do you mind if I just log out and log in a second and share my screen? So I'll be right back.
Okay, so then if there is another question for Dan, we have 1 more minute. So
yeah, sorry about that. I should be able to. Oh yes, now it works. Sorry, I've been like troubleshooting a little bit, but it doesn't. Yeah. Okay, I'll share my screen. Can you guys see my screen?
yes.
Yes. And so let me also put in the Zoom chat a link to the paper, if I can. Yes. Okay. So I slightly changed my title because I realized that I wouldn't be able to talk about everything I want to talk about in the short time. So do I have, like, how much time do I have? Oh, Tilo, you're muted. Sorry.
20 minutes plus
Thank you.
a little bit time for questions.
Perfect. Okay, so today I would like to talk about a recent work that we did with Merav Stern, that many of you know, and Nico Strate, who's the first graduate student to defend from my lab. And this is about how a big heterogeneity of timescales can arise from chaotic dynamics in random neural networks, and this reveals like a new kind of metastable phase of chaotic dynamics that we're very excited about. And so before I start, I first want to highlight the people who did the work in my lab. And so this is the lab, and the people who did the work are Merav Stern, who's a Hebrew now, and Niku, and this is our preprint that's now under revision, and I encourage everybody to look at it if you want to know more about it, about the details. So the kind of work that we do in the lab is about studying temporal dynamics and temporal organization of behavior and neural activity, and so we look at this relationship between behavior and neural activity in both kind of incoming sensory input that drives activity in sensory cortex, and also outgoing motor activity that generates behavior. And we're interested in this kind of the temporal organization that can be kind of approximately organized in terms of three different axes of temporal variability. The main thing we want to talk about today is the hierarchical structure, such that many timescales are present in behavior at all times. So from 100 milliseconds to seconds to tens of seconds to minutes to hours and years, even for humans. Although we study other directions of variability, which is contextual modulations of these timescales, and how at every level of this hierarchy, temporal hierarchy, there's like an inherent stochasticity in behavior that arises, we believe, from metastable dynamics. And so we study the neural mechanisms of these different these different temporal variability axes. And in particular, today we're going to focus on how a hierarchy of timescales might arise from some simple recurrent neural circuits. And in particular, we will see that we will propose that in order to generate neural activity that simultaneously vary on many different timescales, orders of magnitude apart, the simplest mechanism we can think of in terms of emergent collective activity is the activity generated by neural clusters of different sizes. So this is going to be the punchline of the talk. And so why are we interested in this? Well, there's this kind of series of observations from experimental studies that point to large heterogeneity of timescales across cortex. So if we think about the autocovariance function, the spike autocovariance of single neurons, This is what I showed you here on the left. You can estimate the time scale, the typical time scale in many ways, as the full width at half max, or in the case of this paper on the right from the Xiaojin Wang's lab, in terms of the average decay time of population activity. And what they found in the monkey cortex, awake monkey, is that the average timescale of population activity, multi-unit activity, varies along the cortical hierarchy from very fast timescales in the kind of visual stream and the occipital cortex to kind of much lower timescales as you move towards the frontal cortex. And they vary from 50 milliseconds to 350 milliseconds. So this is the average timescale of autocorrelation within different areas. What's even more striking is the fact that if you focus into one single area, now, for example, the anterior singular cortex, and this is work from Johnny Wally's lab, what they found is that different neurons within the same circuit have very, very different time scales, again, measured from their autocorrelation during spontaneous activity, that vary from, let's say, 10 milliseconds all the way to 1 second, which is the longest time scale they were able to measure, to estimate in their data set. But in our hands, it extends even to longer time scales. So this huge heterogeneity of time scale is seems to be a crucial feature of brain activity that reflects also the structure in naturalistic behavior. And so our question is, so what's the simplest mechanism that could give rise to this heterogeneity of timescales in terms of recurrent dynamics? And so kind of a fun, a simple model to study this from first principles, if you want, is a network of recurrently connected rate neurons, rate units. And so that's what we started from. And so our point of view here is a phenomenological approach in which we think about every unit in this recurrent network has a population of neurons. So we think about this as kind of a phenomenological model of some cortical activity. Every unit represents like the average activity in a mean field sense of a bunch of neurons that have the same functional properties, for example, some kind of a Hebbian assembly. And so I wrote here on the bottom left the usual equation for the dynamics of these networks. And so UI is the activity of one of these neural clusters. And they're all coupled together with recurrent couplings, and they're usually taken from Gaussian distribution with mean zero and variance G squared over n. This is the usual parameterization of a random neural network. And the extra ingredient we're gonna focus on is a self-coupling term that you see here that we call S, which in our network is of order 1. As opposed to the usual kind of Crisanti formulation where this is order 1 or square root of n. And so S represents, roughly speaking, the product of the size of this neural cluster times its own recurrent coupling, if you want. And so this is a model that was proposed by Merav several years ago, and it's kind of super interesting in its own right. But what we're interested in today is the autocorrelation to study the autocorrelation function of this model. And so this model has like an interesting dynamical picture with different phases. And there's one phase in which this model displays chaotic activity that I showed here on the right. So these are 3 example units from this model. And so if we take the connectivity such as here, in which the off-diagonal parts, again, are these JIJs that are taken randomly from this Gaussian distribution. And we can fix the self-couplings. So the self-couplings are the diagonal in this coupling matrix here, in this connectivity matrix. We can start from a simple example in which all the self-couplings have the same value. Here, for example, is like 3. And what we find is that we can measure the autocorrelation of all the neurons in the network, of all the units, sorry, in the network. The units represent these neural clusters. And what we find is all the units have the same autocorrelation. There's no heterogeneity of timescales here in this network when all of the self-couplings are equal. And so this is the autocorrelation of the activity, and we see that the histogram across neurons is very, very tight. So there's no heterogeneity. Okay, so this doesn't, so the simple network like this doesn't work, so doesn't have any heterogeneity of timescales, which is what we were what we're trying to model. And so, well, you guess where I'm going. So now what we take instead is we take now different self-couplings for different subsets of the units. Let's say half of the units have one particular self-coupling. The other half of the unit has a different value of the self-coupling. For example, this top left half have they have self-coupling equals 1, and the bottom right part has self-coupling equals 4. And now we can calculate the autocorrelation function of all the units with one self-couplings and all the units with the other self-coupling. And what we found is that, lo and behold, this works. So a network with where units have different self-couplings produces different timescales. And so in particular, the timescale of one unit depends on the self-coupling, on its own self-coupling. So in this case, we have two groups, one with a small self-couplings that has a small timescale. We define the timescale as the width a half max of the autocorrelation function, if you want. And the other population with a large self-coupling has a large timescale. So this kind of works. So this idea then that neural clusters of different size generate different timescales simultaneously within the same network. And we can understand all of this, all of these dynamics in this network in terms of dynamic mean field theory, if you want, where we replace here the input to a particular unit coming from the recurrent couplings with all the other units in the network with a Gaussian mean field, and we can write the self-consistent theory for this. And what we find is that, well, so this theory has a lot of other interesting features. For example, as a function of the two self-couplings in the two groups here of neurons, we have a complicated phase diagram where in some regions we have chaos, and in some regions we have transient activity with fixed points. I just wanted to give you like an example of what this looks like. So if the self-couplings are one self-coupling is small and the other is large, here in the region 1, we have transient activity with kind of the small self-coupling population fluctuates around 0. The large self-coupling populations fluctuates around large values. And yeah, there's a very cool phase diagram, but I'll skip it for now. And so what's interesting is that we can focus on these chaotic activity regions, because that's what we want to look at. We're interested in this autocorrelation function in the chaotic regime. And if we fix one self-coupling to be S1 equals 1, if you want, this one, and if we increase the value of the self-coupling in the second group, then we can calculate the ratios between tau2 and tau1, the self-couplings in the two population, and we see that this ratio increases exponentially as a function of this ratio of the self-coupling. So there's a parametric separation of scales in this network that we can tune by changing the ratio of the self-couplings. So okay, so this generates two self-couplings, but we want to have some network that generates the full heterogeneity of timescales that was observed in the data by Kavanaugh from the Wallis lab.
And 4 minutes roughly.
Oh no, 4 minutes only. Okay,
okay,
stop.
5.
Okay, cool. I go faster. Okay, so how do we do that? Well, we take a distribution of self-couplings. For example, we take a log normal distribution of self-couplings here on the top, and what we find is that we find the long tail distribution of the log of the time scales, right? So which kind of matches this heterogeneous distribution of autocorrelations function from the Wallis lab. So okay, so where's metastable activity in this game? So what we found is actually something very interesting, that when the self-coupling becomes very large, then the network activity, so what I'm showing you here is a snippet of network activity for a unit that has a very large self-coupling, equals 5, for example. Then the network activity becomes metastable. So the activity is localized around 2 values, plus or minus the value of the self-coupling. And it has these very long-lived bouts where it's localized and it fluctuates around those values. And then it abruptly switches to the other value, and so on and so forth, such that the distribution of dwell times around these plus and minus S2 values is very long tail. And so we can study this particular regime, again, using mean field theory in the probe approximation, when we take one neuron with very large self-coupling and all the other neurons with the same self-coupling S1 equals 1, if you want. So we have an effective theory for this probe neuron, which is described by this kind of Langevin equation, where the noise is the Gaussian mean field that has a particular autocorrelation function, which is the average autocorrelation function of all the other neurons in the network. So it's kind of a Langevin equation driven by color noise. And okay, you might think, okay, we have a Langevin equation. Let's study the Fokker-Planck description for this to capture the distribution of values and also the transition times. And it turns out that if you use the naive Fokker-Planck equation that you will get from this, but with white noise, where this was not the actual self-consistent mean field, but this was just the white noise, then this doesn't work. So what I drew here is the histogram of the values of this of the activity of this probe neuron in blue, and overlaid with the solution to the white noise Fokker-Planck equation, if you want, and you see that it's completely wrong. It doesn't work. So in dashed line you see the potential, which is the integral of this blue box, and with the red you see it doesn't work. So it turns out that there is what's called a universal color noise approximation to the Fokker-Planck equation, which is an extension of Fokker-Planck that that allows to consider color noise, so noise with a non-trivial autocorrelation function. And you can actually use this universal color noise approximation to calculate a correction to the effective potential in the Fokker-Planck. And what we found is that that works very, very well. And the effect of the color noise, which is the mean field of all the other neurons in the network, is that it shrinks the distribution of activities that you will get with the white noise. And so this is the full exact description with the universal color noise approximation that you are very excited about, because it's very different from the white noise. And we propose it as kind of a new interesting way to think about recurrent network dynamics, if you want. That captures this metastable phase. And from this Fokker-Planck, we can calculate the mean first passage time between these quasi-stable states in this metastable regime. And it turns out that we can calculate it analytically, and we find that the logarithm of the average first passage time goes with the square of the self-coupling. So this, the time scale of these neurons is exponential in the square of the self-coupling. And for large self-coupling, it matches very well the simulations here, and the universal color noise approximation kind of goes well. And so okay, 1 second. Okay, 2 minutes. I'll finish this. We show that this captures the dynamics of now spiky networks of excitatory inhibitory neurons arranged in cluster, where if you do the mean field, you find that a self-coupling arises from the recurrent interaction of neurons within the same cluster. And if you now simulate network of EI clusters with a hierarchy of cluster sizes, you find again here on the y-axis that the log of the average dwell time is quadratic in the self-coupling. So this matches the qualitatively the results from recurrent network dynamics. Okay, so something else. Okay, I don't have time to show you, but it's in the paper. What is this good for? Well, we can use this this network with multiple timescale to do a demixing of broadband inputs using this idea that Kanaka-Rajan had of suppressing chaos. But yeah, I guess I don't have time to show you that. I can show you if you ask me a question. So let me wrap this up. So again, this is work by Merav and Niku. So we have a distribution of self-couplings that generates chaos with a heterogeneous distribution of timescales across clusters. In this particular limit of large self-couplings, we recover a metastable regime where we can study with this correction to the Fokker-Planck theory. And this reproduces this kind of metastable dynamics that we studied in the lab for many years, which is this kind of cluster excitatory inhibitory spiky networks. And this is a cool application in terms of the mixing broadband inputs. And yeah, that's it. I can stop here. I'm super over time. Sorry about that.
No, that's okay. You just started later, but there's a plenary talk also coming next. That's why I was a bit rushing. Yeah, thank you very much for this very nice talk. Maybe there's time for a brief question. So I don't see one. Yeah, so if there are some more questions, you can stay on maybe a few minutes. But I will then formally close this mini symposium because there is the plenary talk now starting and in channel 1. Yeah, so I would like to thank again all the speakers of this mini symposium, and yeah, it was very, very interesting. Thank you very much.
Thank you, Tilo.
Thank
Hi,
you.
everybody.
Thank you.
Yeah, so